Good evening, everyone. My name is Christopher Baudois, and I am a master's student in the Bioscience Division. And tonight is a true privilege for me to introduce our keynote speaker, Professor Cindy Vandover. Here at KAUST, we focus on pushing the boundaries of the unknown in science and engineering. An explorer at heart, Professor Vandover embodies this idea. She is currently the Harvey W. Smith Professor of Biological Oceanography at Duke University and is one of the leading experts of biodiversity and conservation biology in the deep ocean. In her lab, she primarily investigates the exotic physical and biological events in hydrothermal events thousands of meters below the ocean surface. Her academic work has been featured in Science News, Discovery Magazine, the New York Times, and the US National Public Radio. And although I could speak for hours on the awards and recognition that Professor Vandover has received for her academic work, she is also a celebrated science communicator. She has authored a popular book describing her adventures as a submarine pilot in the deep sea and has been the curator of a traveling art expedition depicting the beautiful sights and sounds of the deep ocean. Her interdisciplinary methods of both approaching and describing science put her at the leading edge of today's conservation scientists, where she also advocates for the ecologically safe use of seabed mining. So, if you've ever wondered what creepy creatures we find at the bottom of the deep ocean and how we can find them, please join me in welcoming Professor Cindy Vandover.
Thanks very much. Thank you. It is really a privilege to be here and an honor to talk with you and to be part of this winter enrichment program. So thank you very much for this opportunity. And thank you to the Red Sea Research Group and Carlos, especially for hosting us and all this wonderful hospitality. I thought I would try to tell this, to give this talk as a narrative. Maybe you'll see the narrative, maybe not, a little bit of storytelling. And so I'm going to start, surprisingly perhaps, with some of the early ways of looking in the deep sea. So Alexander the Great, 300 years BC, would climb into a glass diving bell to explore what was under the surface of the sea. But when you look out here at the Red Sea, how many of you in your minds drain it and think about what it looks like? That's what I love to do, and that's clearly what Alexander the Great was trying to do. Is like, what is it like down there? What's under the surface? So that's what's fascinated me and motivated me in a lot of my work, all of my work. The deep sea, we jumped from 300 BC to 1823. There wasn't a whole lot happening in deep sea research in the intervening years. And you can see this in at the time, what was the equivalent of the internet, World Wide Web, Encyclopedia Britannica. Through want of instruments, the sea beyond a certain depth has been found unfathomable. So that's the situation. That's what we have to deal with. How do we access the seafloor? And we're getting better and better and better at it and discovering all kinds of wonderful new things. However, not everybody believed there was life in the sea, on the seabed. So Edward Forbes in the 1800s, mid-1800s, said there's no life there, it couldn't possibly exist, the pressure is too high, it's too alien environment, there just cannot be life, despite some evidence already that there was life in the deep ocean. And so that was really one of the impetus for the Challenger expedition, which was a expedition led by the British, run by the British, to go around the world and sample the deep sea. And you can see on the right, the deep sea sampling gear, so a gear that's hung over the side, dropped all the way down to the greatest depths of the ocean to pick up animals, brought them back. The animals, and maybe marine scientists here will have seen the animals that come back in a trawl net. They're really in pretty rotten shape. It's not a pretty thing to pull something up from the bottom of the sea in a trawl. But they did. They learned a tremendous amount about life on the seafloor and inspired the next generations to continue working there. So these trawls, trawls and dredges, things hung over and kind of dragged blindly over the seafloor were state-of-the-art for a long time. And then in my narrative, because I'm an American, you're going to hear a lot about the U.S. assets. If you could go to other countries and hear similar narratives with other kinds of vehicles. So forgive me for being Americentric, but this is what I know. So in the 1930s, this might be worldwide, the first person to go half a mile down was William Beebe. And his vehicle was really, so I was an Alvin pilot, so a submersible pilot, so I'm kind of a vehicle snob. And so we call this a dope on the rope, because it has no thrusters, it's hung on a cable, so it's never let free, so it just goes down. The scientist is inside and can observe through the viewports, can communicate to the surface, but can't drive. He's not really a pilot, he's an observer. So he was brave to go in this thing, so I give him all the credit in the world. And it's wonderful that he had this sphere. And you're going to see a similar sphere in many of the vehicles in all the vehicles, except one that I can think of, that we use to put humans on the seafloor. So he didn't really spend time on the seafloor. He was in the water column, and he was what made some of the first observations of the midwater pelagic fish and other creatures that live in the water column. Very exciting work. We also had early on dives to the Challenger Deep, so greater than 10,000 meters water depth. And this was the bathyscaphe Trieste, operated by the U.S. Navy. This was where two people, Don Walsh and, I forget Picard's first name. Anyway, Walsh and Picard, went into, you can see that sphere again, so sphere with high pressure on the seabed. You want to be in some kind of non-compressible unit, so a thick metal sphere. You can see on the left there's a viewport, and then there's a huge tank that was filled with gasoline for buoyancy. So gasoline is lighter than seawater. So that was the buoyancy. Then you need to be heavy to sink. So you see on the bottom the two cones and dispensers. That's where lead shot was stored. And so they'd fill it up with lead shot. The vehicle would sink to the bottom. So they weren't driving, there's no propeller, no thruster, but they could go up and down, so without a tether, without a cable. So they could go down, and then when they wanted to come back up to the surface, they'd drop the lead shot, and that gave them their buoyancy to come up. So they made a successful dive to the bottom, deepest part of the ocean. They were there for about 20 minutes. The light imploded while they were down there, so they had very few seconds of actually observing anything. And I've talked to Don Walsh, and mostly what they observed was a cloud of mud that came up when they hit the bottom. But anyway, they got there. So that was more engineering, record-making. And then we had another record-making person, James Cameron, the film producer, who built the Deep Sea Challenger. Really remarkable, state-of-the-art. This is the only vehicle you'll see that's in a vertical orientation for speed, for getting down to the bottom of the sea. So we went down to the bottom of the Challenger Deep and made some observations, picked up a few samples. But since then, nobody else has been back down. It's probably not going to be any humans down on the Challenger Deep for a long time. Jump to 1964 in the US, and we built our first research human-occupied vehicle. This is Alvin, and this is the vehicle that I piloted, so you'll see a fair amount of Alvin just because I love her so. And she has, you can see the personnel sphere, so that's where the people go. In this case, she can carry 3 people, and she's always died with 1 pilot and 2 observers, I think always. Anyway, as long as I've known her, 1 pilot and 2 observers, which is different from most of the submarines in the world that have people in them. It's usually 2 pilots and an observer. So we kind of, another snobbery that's proud of being the solo pilots in a submarine. She is buoyant for a couple of reasons. There is a buoyancy pack. I don't have a pointer, so you're just going to have to kind of follow my directions. But, well, let's start aft. There's a propeller. So there really is a pilot. The pilot is not tethered. Pilot can drive and move forward and aft, up and down, sideways. So the propeller is on a pan and tilt, so it could move and have directionality. Then if you go forward of the propeller and rudder, there's buoyancy material, that big pack. The buoyancy material, I think that was code, because they didn't want to tell the world at the time in 1964 that they had developed this kind of buoyancy called syntactic foam. And so it was kept a secret for quite a while. So syntactic foam is little microspheres, glass microspheres embedded in epoxy. So in air, it's very heavy, but in water, it's buoyant. So that's what gives the vehicle buoyancy to come up. And then there's variable ballast tanks and high-pressure air that are used to do the neutral trim. So if you're driving over the seafloor, you don't want to be just kind of having to go up and down. You want to just stay nice and calm. So that's what those are for. And then you see the personnel sphere. Then there's also a main ballast that's just you blow the vents and the sub sinks in the morning because you've hung weights on it and you've gotten rid of the air. She has cameras, viewports, you know, kind of a good old submarine. So this is the basic model that every submarine that exists now, except for that deep-sea Challenger, is based on the human-occupied vehicles. They used to be called manned submersibles. So Alvin has done a lot of famous things, including, especially when she was a young kid, she did some interesting things. One was the famous project, so the French-American mid-ocean ridge undersea studies. And she went to the Mid-Atlantic Ridge to explore what's there. So you're going to hear a lot about mid-ocean ridges, Mid-Atlantic Ridge, mountain range girdling the globe, bisects the Atlantic Ocean. It's a spreading center, seafloor spreading apart there. And it says here, as with the Apollo project, extensive unmanned surveys are being conducted before men are sent into this region, whose landscape is more alien to human experience than that of the moon. And that's still, it gives you goosebumps. It's still true. It's an alien place. And so this is a little picture of how they did this. They had a towed camera vehicle that they towed over the ridge axis, sometimes in a slalom shape. This was back in the day of film, right? So they took the film, developed pictures, laid them out on a gymnasium floor to plan where they would do their Alvin dives. It's a little bit of a painful way to think about these things, given the technologies we have now. But clever. Human-occupied vehicles, you're at risk. You could die. Alvin sank. Nobody died. There were 3 people in her when she sank. They got out. She was on the surface and they got out. But the submarine filled with water and sank to the seabed and was there for 10 months. And I tell you this not just to make you remember it's not a place, it's like going into outer space, right? Not a place humans are normally found. But to tell you the story briefly of the bologna sandwich. So if you're a microbiologist, you might have heard about this for deep ocean. When they brought Alvin up, they pulled the sandwich out. You go down with a sandwich and an apple and maybe some soup or something. They pulled the sandwich out, and can you see it's a copy of a copy of a copy, but there's a bite out of that sandwich. So it's bologna on, if you're American, you know about Wonder Bread, which is like the most preserved bread in the universe. So Wonder Bread and bologna. And the microbiologist in Woods Hole, Holger Janisch, was struck that there was no decay, that it hadn't rotted. So it sat on the seafloor for 10 months. And so he thought about microbial activity under high pressure and got funding to study this and put down nutrients on the seabed and really helped us understand a lot about, in those early days, about how microbes work in the seafloor. However, if you want to try this experiment, it's really pretty cool. Make yourself a bologna sandwich. Make 2. Put 1 in the air and 1 in seawater in the refrigerator and keep it for 10 months. If you make it with bologna and Wonder Bread, which are 2 of the most preserved materials, foodstuffs in the universe, it won't rot. And you could eat it. It gets soggy, but you could eat it 10 months later without putting it in the deep sea. So a bit of an apocryphal story. Okay, so let's move on to more advanced technologies. Alvin has actually advanced. Alvin still exists, but there is nothing in the vehicle you see on the lower right left from the vehicle in the upper right. So it's been completely rebuilt, and it undergoes overhauls every 3 years for safety reasons, but then it also undergoes major renovations, if you will, every so often. So one of the big renovations was in 1989, a big rebuild where you can see in the 84 picture, there's a propeller. The propeller gets lost. We move to electric thrusters. So in 1989, I was finishing my PhD degree in Woods Hole, in the joint program, Woods Hole MIT program. And I had already been working with Alvin. I'd had a couple of dives in it as a graduate student. And what I saw was, if you're an ecologist and you want to understand the seafloor and deep sea environments, especially hot springs, you want to be an Alvin pilot because they dive day in and day out. They go all over the globe. They know how to use the submarine really well. So I said, I want to be an Alvin pilot. And that was not an obvious thing to do. but I wrote a proposal to the National Science Foundation for a postdoc to do it, and I got some really awful attacks on, who are you to be a submarine pilot? But I got in through the back door. They were taking it apart and rebuilding it, and the US Navy required that they write a maintenance manual. So I said, I'm writing my PhD dissertation. I know how to put long pieces of writing together. So I would go around following the Alvin technicians as they rebuilt the submarine, actually as they took it apart and put it back together again. And I wrote the maintenance manual, the first draft of the maintenance manual, including all the computer diagrams and all the checkoff sheets. And so I got to know the Alvin from inside out. And then I was invited to join the Alvin group as an electrician, of all things. So I worked with Alvin for a couple of years, first as a pilot in training in Alvin Pitt, and then as a pilot in command. So then I went back to science in my academic career, but in the early 2000s, we decided to make a newer, better, deeper diving Alvin, and so that's what you see in kind of recommissioned in 2013. So it's capable of going to 6,500 meters now and has just much more modern capabilities. And I was lucky I got to be chair of the oversight committee. So as it was being built, I saw all of the engineering that went into all the decision making, and we had scientific input into how the submarine was built. It's a very interesting adventure. This is what Alvin looks like now. So here she is on the left in her hangar on the ship. She requires a mothership. She can't go off of any ship but Atlantis. So it's a little difficult. She goes around the world. She can work in the Pacific, Atlantic, through the canal. But you can't put her in a plane unless there's a disaster and she needs to go somewhere. Then she can do it, but not for science. Inside, she's very complicated. One of the things I was told to do and did do as a pilot in training was go inside the ball, the sphere, and shut my eyes and touch the different switches to do a particular maneuver on the seafloor so that you could do it all in the dark in case you needed to. And just really to be very efficient at moving moving the submarine. In addition to human-occupied vehicles, we use a lot now the remotely operated vehicles, so we saw some of those in the afternoon talks. This is Jason, the one we have operated by Woods Hole. This is a tethered vehicle. It's much smaller than Alvin, no human anywhere near inside of it, but it's almost as capable as Alvin. It can't carry big, heavy loads, can carry a pretty respectable payload of science samples, and has really good video capabilities as well. So I use that a lot. And in fact, I prefer using the ROV now. It's much more than using the human-occupied vehicle. So give me a choice, I'll take Jason over Alvin any day. One of the reasons is Jason is a very democratic vehicle. Everybody can see what's going on. With Alvin, it's very elitist. Only the two scientists who were in the submarine and the pilot pretty much know what was done on the dive that day. There's no routine communications to surface except for using a Morse code or very brief communications. You really have to push. It's not, the culture of the pilots is not to talk to the surface. They just want to work. You're only down there for you have an 8-hour dive, so you have 5 hours on the bottom typically. But with Jason, you're up here in a control van. So you have all the videos that you can watch. There's multiple views. The pilot is in the foreground here. The chief scientist sits right next to him and whispers in their ear and tells them what they want done. There's a navigator, there's an engineer on the other side. And then the back row of this van has the scientific students and collaborators. And so it becomes a joint effort. Everybody's involved. And there's repeater video screens in the main lab, in some of the cabins even. You could watch what's going on and run up to the control van when it's time to get your samples or when you see something interesting. So it's a very democratic way of doing work. My new favorite toy, and this is because it's just so cool, Sentry. So Sentry is an autonomous underwater vehicle. It's cool because it's so capable. It can do amazing things. So for me, I like to map. I'm an ecologist. I want to know where my animals are. It's hard to piece that together in a submarine. Even with an ROV, it's hard to, with a ROV, when you have a human factor there, it's hard to be regimented and just drive survey lines, right? You see something and then you veer off and you mess up your survey. But with this, you program it, send it over the side, it has no tether, and it has no choice but to do what you told it. You have some limited ability to command it and have it change its plans, but mostly the heavy-duty programming is done before it goes into the water. So it can go down to the seafloor, it can make very high-resolution bathymetric maps. In fact, so high, in one place I was working, admittedly, the mussels were really big, but it could image those mussels just with sound, so sonar. So it has very high resolution mapping, it has side scan sonar, and then you can, even on the same dive, you can do your high-level map and get your basic bathymetry, and then if you already know there's a particular place you want to go and get high-resolution photographs, a photo survey, photo mosaic, you can just drop down the same dive and do that. You can go over to some other place, do some activities. So you have a lot of time, a lot of good survey tools. And at the same time, it's taking the pictures or the bathymetry, it's collecting sensor data. So you can put any sensor on it that you want. It has the capacity to record lots of things. So one of the fun things, and I've talked about this with a few people, and I think it's just true of engineers everywhere. When you get to go out to sea and work with engineers, you can do fun things and push the state of the art. So one of the things we did recently was I was working with a big project. We were trying to understand connectivity, genetic connectivity among seeps from the Caribbean into the Gulf of Mexico and then up off the Atlantic margin. So this is looking at mussel larvae, really, and some worm larvae. So we had my team from Duke was looking at the genetics. We had a team from University of Oregon looking at the larvae and where the larvae were in the water column. Then we had a North Carolina State University group that was looking at the physical oceanography. So where could those larvae go? And did the genetics match what we could see with the larvae? The larvae guys use this thing called a mock nest, multiple opening closing net environmental sensing system, which in the 1970s was an amazing tool. And it has been the workhorse. It's still a really good tool to use for sampling in the deep sea for zooplankton. But you tow it behind a ship. It can sense oxygen, temperature, salinity, but you have to tow it obliquely to get a sample, and you can sample intervals. It has 9 nets, so you can drop one net, open it, drop another net, close that one, and you open another one. And so you can sample depth intervals back up to the surface. You can't put it down a meter above the bottom, because you don't want to crash into the bottom. So you're really working 50 to 100 meters off the bottom. What's down in that lower 1 meter above the bottom? That's where all the larvae are. So that was one of our problems. We wanted to get a meter right above the bottom, and we wanted to sample large volumes of plankton, and we wanted to do it with precision. We wanted to know where the samples came from, and we wanted to have a chance to really push the envelope. So we went to the engineers. The Sentry was on the cruise the same time we were using this. This would also come up every night. I'd go to bed, and the larval team would take this thing, and they'd work hours lowering it down to the seafloor, bringing it back up, cleaning it up, and it would just fail over and over again. It was just awful. So let's build a better mousetrap. So this is what the engineers came up with. I can't take any credit for the design, but it was the specifications. Here's what we need. And then the engineer said, oh, well, okay, here's how you could do it. So this is a new design for a plankton sampler. It's a tube within a tube, so the filter tube to collect the larvae, or zooplankton. At the aft end, there's a pump. And so the water is drawn in through those cones and moves over the filter. And then at the cod end, just like in a cod end of a mock nest or any plankton sampler, there's a capture screen. And they designed it. They knew also the larvae had to be in really good shape, or the zooplankton had to be alive when we got them back to the surface. And so they designed a nice cod end with a little chamber that would capture them and keep them soft and gentle. Very large volume of water throughs here. It had to match the volume that would go through a typical mock nest tow. So that's what this can do. And then when it's mounted on Sentry, that's what you see here. We had a scuba diver go underneath it one dawn and took this dramatic picture of this thing. It worked out of the box the first time. It was just extraordinary. So it can take 2 samples on a mission, which isn't that much, but they're really good samples. The quality of the larvae is exquisite, and the volume of water sample is great. The larval biologist had never collected so many larvae in his life. So he, from being a cynic and a critic, he became an enthusiast, which was great fun. So that's just from having been on the boat with the engineer and saying, well, here's what I need. And so he figured out how to get it to me. The other thing we're doing now, that I absolutely adore. I love to go to sea, but it's pretty cool if you can have a ship out at sea and a team also working on shore. So this is called telepresence. And Bob Ballard in the U.S. was one of the leaders, was the leader for telepresence. He uses it a lot all the time for outreach. So you get school kids to go into an auditorium in museum audiences, and they get to see the ROV video feed coming up to the surface and satellite beam to them, and they can engage with the science. And there's even scientists who work with those programs. But I was really interested in kind of pushing the envelope on this. So you'd think, okay, telepresence, you have ROV feed, you've got video, you're not going to get bored. But I didn't have an ROV on a particular cruise I wanted to take it out on. I had Sentry. Sentry goes down, it's not live data. So we had there's an engineer there who was on shore. He would work with us on shore to program the vehicle. They did everything but the final checks, basically, from shore. It was kind of a proof of concept. Could you do it from shore? And we did. And so he would send the program out, and then they would put it over the side, and it would go do our mission. We'd go to bed, and then in the morning, they would be feeding the data back to us by satellite. And I had this army of students who we just sat at the computers, worked up the data. We had our paper written by the end of the cruise, because there was nothing else to do. It's like being on a ship. You're trapped. So we trapped them. We pretended like we were on a ship. So it's a very interesting way to do science. I did it again with a group of early career scientists. We had 12 scientists on shore, 12 on the ship for a week each, and then they swapped and they each had the telepresence experience. So it's a very powerful way. We haven't really learned how to use it fully, but it's pretty exciting. Okay. I want to tell you a little bit about some of the organisms that live in the sea, or the habitats and diversity. I think this is a pretty sophisticated crowd, so you know there's a lot of habitat diversity out there. But I've picked some of my favorites, and especially because if you're not in marine sciences, you might be thinking, I don't know why I care about the bottom of the ocean, but I don't. So a lot of people just don't know why it matters. So I'm going to try to tell you a little bit. So we were doing some recent work off the canyons in the Atlantic margin of the U.S. It turns out there's 500 seeps that were discovered just in the past couple of years. There's seeps. Seeps, what are seeps? Seeps are places where they're like oases of life on the seafloor. So we wanted to study these seeps. You'll see some more pictures. It'll become clear what they are. On the canyon walls, there are beautiful animals. So the NOAA Exploration, our National Oceanic and Atmospheric Administration, has been doing a lot of ocean exploration, especially in these canyons, and really shown the beautiful cold water corals that exist there, very high diversity. And so these canyons, several of them have now been protected by President Obama. They may not be protected next week. And then there's whale falls, and I just wanted to introduce you to this lovely worm. Somebody told me this morning they didn't think it's like too early in the morning for worms, but to me it's never too early for worms. These are the bone-eating worms. So when a whale or a big lipid-rich skeleton falls to the seafloor, the meat gets scavenged by fish and crabs and amphipods and other creepy crawly things, brittle stars. And then sometimes, often, the bones get colonized by these bone-eating worms. So you see the red on the bones themselves on the vertebrae. Those are the little worms, like you see close up, you see tentacles. And then off to the right is an individual photo that's probably by Greg Rouse. He's an amazing invertebrate photographer as well as invertebrate taxonomist systematist scientist. They don't have a mouth, they don't have a digestive system. They do have this root mass at the bottom that grows into the bone and slowly consumes the lipids that are in the bones. They have bacteria inside of them. And all these guys that you see, all that red, they're all females. The male is microscopic inside the female, so you can't see the males. Brine pools, we saw some brine pools this afternoon here in the Red Sea. These are ones in the Gulf of Mexico, and a little self-portrait of a different ROV that is run by the University of Rhode Island. So these are lakes on the seafloor. Very peculiar thing to see. It's really lovely. And they have a shoreline, they have a beach. In this case, we see beds of mussels on the top picture off to the right. Very high salinity fluids in the brines, obviously. And then we also see the methane bubbles. So this is a type of seep. Methane is seeping out of the seafloor, rising up, going into the water column, feeding lots of bacteria. The hard substratum there is orthogenic carbonate, which is made by microorganisms. Pretty amazing stuff. What's certain is we haven't found everything that's strange on the seafloor. Okay, I want to spend a little bit of time on hot springs, so that means we have to talk about mid-ocean ridges. So the mountain ranges that girdle the globe, again, sorry about not having a pointer, but in the eastern Pacific, there's the East Pacific Rise. In the western Pacific, I'm going to talk about a place called Manus Basin, or in the Bismarck Sea, which is just north of Papua New Guinea, which is north of Australia. And then, let's see, we'll see on the Mid-Atlantic Ridge, we'll see some things. And then I wanted to remind you that the Red Sea is mid-ocean ridge. So this off to the right, that little valley you see there and ridge system, that's the central Indian Ridge coming up, going into the Gulf of Aden. And then there's this incipient seafloor spreading, so the Red Sea is opening. Maybe you guys all knew that, but I think that's really cool. It's a baby mid-ocean ridge. So mid-ocean ridges, they are formed as the ocean plates get pulled down under the continental crust. It thins the ocean crust at a certain point at the ridge axis, allows the magmatic heat to come up close to the surface. Seawater percolates down into the crust, gets heated by hot rock, gets chemically modified. It strips the rocks of metals and other elements, becomes thermally buoyant, rises up, and forms black smokers. So this is the first black smoker ever seen in 1979, discovered by Bob Ballard and his team. So black smokers look like what they sound like. The temperature at this particular one is 400 degrees Celsius. It's at 5,000 meters water depth, so it is the deepest known black smoker. The temperature right outside that smoke at the orifice is 2 degrees Celsius, so incredibly steep temperature gradient. And then where you see the white reflected area, that's going to be where the 400 degree Celsius fluid is. The smoke is not smoke, it's finely divided metal sulfide particles, and it precipitates to form that chimney. So these create some very interesting kinds of habitats. So if you're not familiar with chemosynthesis, I wanted to contrast it with photosynthesis. So photosynthesis, you all know, we have sunlight, and it's the light energy that chloroplasts uses to make plant material, right? And then that gets eaten by the food web. In chemosynthesis, this is what goes on at hot springs, these hydrothermal vents on the sea floor. We have chemical energy coming out of the hydrothermal vent. Well, it's actually not just the vent. You need the reduced compound, usually hydrogen sulfide, coming out of the vent, mixing with oxygen, and so it's a redox reaction that actually creates energy. And that's done both inorganically, so without life, but also microbes have learned how to do this really well. So the microorganisms do this redox reaction, capture the energy as ATP, and then shunt that ATP into a carbon fixation cycle. The same carbon fixation cycles that we find in plants are used by the microbes that live at these hot springs on the seafloor. And then there's a food web around that. And the really cool thing is these tube worms, which are not the next picture, sorry. So another really cool thing is the discovery of hydrothermal vents in 1979 changed the way we think about where life can exist on our planet, the extremes at which life can live in terms of temperature and pressure. And it changed how we think about looking for life on Mars. So when the Viking lander arrived on Mars in 1976, it was looking for life fueled by photosynthesis. It didn't find it. Now, and for the past, since the late 1970s, NASA has been looking for life, looking for things that would use chemical energy. So it really changed the way we think about not just life on our planet, but the origin of life on our planet and about life on other planets. Okay, here's the promised tube worms. So these are pretty much iconic for hot springs. Anybody who's in marine science has studied this and had to write an essay probably in their exam. They are, well, let me show you the video. So they have this bright red plume, which is red because of hemoglobin. It's a very special hemoglobin. It can bind oxygen as well as sulfide without competition. So it's a happy situation. So the warm water's coming up, rich with hydrogen sulfide. The sulfide is bound to the hemoglobin in the tube worm, in the blood of the tube worm. That red plume is really a gill, right? So it's a very large surface area for absorbing chemicals. The oxygen is in the overlying water, so they're taking up oxygen, and then there's CO2 from both the venting fluids as well as the surrounding seawater. They're living at about anywhere from 10, 15, 20 degrees Celsius. So, well, we have these black smokers. Nothing lives in the black smokers at 350 or 400 degrees Celsius. But these lower temperature diffuse flows, we call them, is where you find all this wildlife. This is not what the typical deep sea floor looks like. This is a mile and a half down, 2,500 meters, and it's just an oasis of life. Actually, if you're wondering about size, the size of this auditorium might be the size of a vent field, maybe the size of a soccer field, football field. Truly exquisite design. These guys, this is a giant worm that can be taller than me and is, kind of big around as that. They have no mouth, no digestive system. And what they have is a sac full of bacteria inside of them. So they have to move the nutrients down to those bacteria. So they're taking up hydrogen sulfide, which is normally a potent toxin. The reason it isn't toxic to them is they immediately bind it to the hemoglobin, and then it becomes not available. It's not bioavailable. They're taking up extra oxygen, because they have to have oxygen for themselves, for the invertebrate host, and extra oxygen to fuel all those microbes for that redox reaction to make the ATP. And then they're taking up carbon dioxide. We normally exhale carbon dioxide, but they're taking it up. So they need to take up CO2 for carbon source for their bacteria. Really exquisite design. And then there's lots of animals that live among them, so they're bioengineers as well. Elsewhere, so we have worms that can dominate with the bacteria inside of them. There are crabs, these yeti crabs that live with bacteria on the outside of them that they feed on in very high densities of crabs. Shrimp at very high densities. So you go to different regions of the world's oceans and you find different dominant taxa usually. These shrimp are really remarkable because they have a weird eye. They were described initially as being blind, but instead they've taken their eye, pulled it inside under a thin, transparent carapace, enlarged it, gotten rid of all the optics. They can't form an image, but we think they can detect a gradient of light and that they're very sensitive to a very dim source of light. And if you think about it, you don't know this yet, they live on the sides of black smoker chimneys. So now if you think about it, you can ask yourself, what are they looking at? And you say, black smoker chimneys, 350 degrees Celsius, that's hot. Hot things glow. Maybe they're looking at the light generated by black smokers. There is light generated by black smokers. We went down and took pictures of it, so we know what it looks like. We know its quality. We know the wavelengths. It's mostly long wavelength light, but there's a tail that goes into the visible. So we don't know this, but we think the shrimp are using their eyes to avoid getting cooked by the black smokers. And then there's also mollusks that dominate in some sites. So there's these guys, there's a hairy snail, a black snail, there's mussels that have symbiotic bacteria in their gills. So we find different ways of dealing with the symbiosis with the microbes, but every time we find very high biomass at a hot spring, it's because there's a symbiosis associated with the animal. Okay, so that's the biology, but there's a geology side, of course, to all this. And Jules Verne realized this back in the 1870s. So in 20,000 Leagues Under the Sea, he wrote, "In the ocean depths there are mines of zinc, iron, silver, and gold, which would be quite easy to exploit." Now, he got the quite easy to exploit part wrong because it's hard to access them. But there is metal. There are metal resources on the seafloor, commercially valuable or potentially commercially valuable. So there's three kinds that in my world I think about a lot. The seafloor massive sulfides, which are the rocks that are formed at black smoker chimneys and black smoker systems. There are manganese nodules, which kind of litter the seafloor in certain areas, 5,000 meters deep and deeper, that look like potatoes on the seafloor that could just be raked up. And then cobalt crusts, which occur on seamounts in certain areas. And cobalt crusts, if you want to mine them, they have to be scraped off. But I'm going to focus today just on the seafloor massive sulfides, chat a little bit about them. So here's the location of one on the Mid-Atlantic Ridge. The tag mound stands for Transatlantic Geotraverse. Geologist named it. And I was involved in mapping this site. So this is pretty primitive mapping. This was decades ago. Now we could have much higher resolution maps. But this is what it looks like. It's quite large, 50-meter scale there. So it's a couple hundred meters across in diameter. And here's what it looks like, we think, in cross-section. So there has been some drilling that we know that the sulfide deposit goes down a certain depth, and then there's an altered rock stockwork, we call it, where the hot fluid has come up. And there's also shrimp there. So this is where you find swarms of shrimp. So unfortunately, you have geology, but you also have biology, which can be a conflict. On land, we find the same kind of deposit. Cyprus is a big hydrothermal vent deposit. It was once on the seafloor. It's now been brought up onto land. So we know from looking at land deposits kind of what the dimensions of a good seafloor deposit might be and how many of them there might be. So this is a copper mine in Cyprus. And so ophiolites, before the discovery of hydrothermal vents, how ophiolites formed wasn't known. So now the discovery of vents really changed that too. So here's the metals, minerals of interest, the chalcopyrite and sphalerite and pyrite. So this is going through a cross-section of a black smoker chimney. The chalcopyrite is the hottest mineral. So they're copper and zinc rich, and in some places, especially where there's a continental margin, continental crust influence, you can get gold and silver. And I wanted to remind you that the Red Sea is also a spreading center, so you could have hydrothermal vents. In fact, you don't have seafloor massive sulfides in the Red Sea, but you have metals, you have metal-rich sediments. And in fact, it's the largest known hydrothermal metal deposit in the modern ocean, 90 million tons of metal-bearing muds that are of commercial interest. So there's mining interest. And in fact, my understanding is there's a mining license, so there is a license to mine. The metals, the muds are under the brine pool, and that's about all I know. What from this diagram, what you can see is you have the heat from the magma chamber is making the fluids in the rock buoyant so that they come out and deposit these metals into the mud. And in order to have that convection cell work, you have to draw cold water down. And the cold water is going down over evaporites and shales, evaporites, salt deposits. So you're getting salt-rich fluids going down into hot rock, being heated up, coming out at the seabed, creating brines, so you don't really get a black smoker, you get a different kind of setting. It's pretty cool. The other place where there is a license to mine that's been granted by a country is in Papua New Guinea, Manus Basin, and it's a small Canadian company, first mover company called Nautilus Minerals. So they want to mine there in a place that they call Sawara 1. So Sawara is saltwater in Pidgin, and that's the name of their prospect. Here's why they want to do it. Look at the copper values on the right for the ore quality. So they have the land-based ores are really, what's left is not very rich in copper, but the submarine sites are pretty rich. They're also at 1,600 meters, so they're not so deep. They're in a very protected area, so they could work year-round. So it's kind of a nice test bed for deep-sea mining from a geological operational perspective. Here's what they have figured the deposit looks like. It goes about 30 meters down. They figure it will take about 3 years to extract all the metal once they get started. So far, there is no mining whatsoever anywhere in the deep sea. It's just thinking about mining and having, in a couple of places, license to mine. Here's how they plan to mine. They'd have a surface vessel that sits over the mine site, has a riser lifting system to carry the ore up to the surface. And then on the seabed, they have 3 tools that will grind the seabed, gather it up, collect it, and have it prepared for being picked up as a slurry. The tools are very large. Can you see those 3 people beside the one on the left? So they're quite large. They have not been tested in deep water, to my knowledge, but they're derived from coal mining kind of operations, designs. Well, you notice in this lovely picture, they actually don't show the animals that are there. So there are animals there as well. And Nautilus Minerals, and I think mining companies in general seem to be, we talk a lot with potential deep sea miners, and they're very interested in not damaging the seafloor beyond what is needed to remove the ore. And Nautilus Minerals in particular, as an early mover, came to the scientific community, asked, how do we do this? We understand these animals at hot springs, they're living libraries, they have genetic resources, we don't understand them, they have adaptations we'd like to know more about. We want to make sure we are not going to lose important biodiversity on our planet through our mining operations. They were very proactive about this. So I worked with them to do some baseline studies. It was actually a very selfish maneuver on my part because I was really doing a lot of quantitative biodiversity measures, comparing biodiversity at hydrothermal vents and cold seeps around the world. And so I didn't have access, I didn't have funding to go to Manus Basin. And so they took my students there and we got samples. I would say we probably still have the best, most robust quantitative samples of these systems ever. For when I get funded by the National Science Foundation, I can't take the kinds of samples, the quality and quantity of samples we were able to take here. So we contributed to the environmental impact statement, which was quite comprehensive. It can be criticized, it's missing things, but they're first movers. They're trying to figure out how to do it. One of the things that we were really interested in is understanding what's at risk in these systems, and there's a burgeoning industry among deep sea environmental scientists, this new discipline of people talking about what the potential damage could be. So since there's been no test mining, we don't really know what the potential damage could be. We can't really scale it. So we're trying to take our knowledge and apply it as best we can. Our expert opinion, it's actually a legitimate way of writing papers. You just go talk to experts, get their opinion, and write it down. So one thing to think about is, suppose you do mess up the environment, how do you fix it? We know on land, the mining companies are responsible for rehabilitation. You don't just abandon a mine site, you actually have to close it with rehabilitation, put the plants back there, whatever your particular country requires. And so we asked, well, what are you going to do when you close this mine? And Nautilus Minerals said, well, what do you want us to do? Well, that's a stumper, right? So there is no science of deep sea restoration. And we don't even do it very well for shallow water systems, right? So we can make progress, but we don't really restore back to something that functions the same way it did when it was damaged, or not right away anyway. So we did suggest some things to them that, well, the black smoker fluids are still going to come out. Maybe the animals that live on the sides of the chimneys, the snails in this case, really would benefit from having those chimneys stabilized quickly. So this is the concept of putting down some stabilization platforms over the black smokers that are still there, and seeding them with snails removed from in front of the mining tool. The other thing they're really thinking about is their mining plan, and they just avoid hitting the really rich, dense populations of snails. Maybe they'll die for other reasons. They get smothered. We just don't know. But they're thinking about these things, and we're trying to help them think about it. And then we also think about, well, where do you put a reserve? You want to put your reserve upstream. You want to understand something about connectivity. They funded a very large study of connectivity of, we did 7 or 8 different species within Manus Basin, multiple sites, and then other basins that had the same species. So we know something about that and how the potential for recruitment. So it's been a real opportunity to learn a lot about the seafloor working with a mining company. And the other thing that's, I think might be at risk, I want to mention is cumulative impacts. I really believe in the case of Saguaro 1, if you remove Saguaro 1 and all its animals, it's no worse than a volcanic eruption that might overflow a hydrothermal vent. But that's once. It's when you need to, you can't just stop at one mine. You need to keep going and doing other mines. And so it's that cumulative impact. How will you ever know when you've gone too far? Because we don't really understand how these systems are maintained. And there's some of my colleagues would argue and just say, you know, we shouldn't even do that till we know more. But I'm curious. Okay, so I just wanted to think a little bit about, we talked about 2 national jurisdictions, so the Saudi Red Sea mud mining and the Manus Basin seafloor massive sulfides. And those are in national jurisdictions. What about all this area in international waters? So this is a really interesting place. So this is you know, just intellectually rich, because this area, the ocean, is the common heritage of mankind. And the resources are supposed to be for our generation, but also for future generations. And that's by the United Nations Convention on the Law of the Sea that almost every country has signed on to, except the United States and a couple others. The International Seabed Authority was set up through an implementing agreement associated with the Convention on the Law of the Sea. Seabed Authority is located in Jamaica. Did I say it's set up in 1994? So it's a young organization. And they are responsible for all the regulatory actions related to deep sea mining, mineral extraction in international waters, which is everything in the outside the national jurisdictions. So they regulate mining. They're making all the rules about exploration and mining, but they also are responsible for protecting the marine environment from damage done by mining. So it's kind of a fox guarding the hen house. And right now they're very heavy on, they want to facilitate mining, in part because they need the revenue from mining to fund their business. And the environmental side has been really, they've come again to the scientific community and asked us to help them find their way through some of this environmental management issues. So that's one of the places where I've been very active in recent years. The rules really for exploration and environmental management are being written this year, so we are going to be looking at drafts and commenting on them in February. The Seabed Authority does give out exploration leases, and I'm just showing you here or highlighting the seafloor massive sulfides. So there's exploration leases for China and India in the Indian Ocean, and then in the Atlantic we have a French claim and a Russian claim for exploration. So there's no mining going on, but they're exploring, looking at the resource, trying to figure out where they would mine. And the Seabed Authority has asked us to think about how would we design a network of marine protected areas to put on the Mid-Atlantic Ridge to say, no, you can't mine here. So it's kind of, it's called a precautionary approach to, you know, we don't know what the effects are going to be, but let's make sure we keep a good percentage of the ridge axis safe. And so they have, in other work, the precedent is for between 30 and 50% of the area should be managed as a no-mine zone. So if you look at this ridge axis, we chose to work from the southern edge of the Portuguese extended continental shelf claim, or request, which is the hatched blue up in the north, down to the Ascension Island extended continental shelf claim. And we, of course, the Brazilian EEZ that's in the middle there, we can't comment on. But we had to think about how would you design a marine protected area. And although I'm going to give you some of the answer, you know, be a nice question for your master's exams or something, how you would think about doing this when you know virtually nothing about the animals that live here, right? How do you decide what you're going to protect? So our strategy has been to work with our GIS colleagues in the Marine Geospatial Ecology Lab at Duke, and with an international community. This is by no means me. This is a huge group of people, 40 or 50 people. And we thought, well, this is a linear feature, so our protected areas should have some kind of a linear array. And that captures the latitudinal gradients that might exist in productivity and particular organic carbon flux to the seabed. It captures some bathymetry. There's not so much change in bathymetry as you go down the ridge axis, but along the ridge axis. So we're thinking about we need to have protected areas that go out to either side to capture the representativity, to have representative areas protected. So we think about, we want 30% of the fracture zones, all those horizontal lines that go across are fracture zones or transform offsets. They're a little bit deeper, so we want to capture 30% of those. There's seamounts in here, so we have maps of seamounts, published maps, databases. We want to capture 30% of the seamounts. We want to capture, we can take the digital data and plot the depth distributions, and we want to make sure the depth distributions of the areas we set as no mining match as best we can, or a reasonable match to the full set of depths that are represented and in the right proportion. So we're playing this game, plotting all these things, thinking about where to put these boxes. We also think about the biology of the invertebrates from analog taxa. So we know some things about dispersal distances. And so we take that into account. You want to have a box that's protected, that's big enough that the populations within it could be self-sustained, that they disperse just within that box rather than outside the box. And then minimum spacing between boxes. So I can't show you the answer because we haven't told the Seabed Authority what we think the answer is. What will happen, though, the process is really interesting. We had initial meeting where we said, how are we going to do this? And we kind of came up with a sketch of what we'd like. And then we had a smaller group that really worked with the data and said, okay, here's how it could really work. And then we came back together as a larger group to build consensus, make sure we're on the right track. And now we have a smaller group that's gone back to kind of refine it. Eventually, we'll get to having a publishable paper that says, here's our science-based recommendation. Now, the Seabed Authority, reasonably so, isn't only responsible for the science, they also have to facilitate the mining. So they'll work with us, we'll engage with the Seabed Authority, and they will work with contractors, the mining contractors, and there'll be give and take, and we'll figure out how to do this together. But we wanted to just have a chance to work independently. Here's what, as scientists, here's our recommendation given what we know. So that's how we've been handling this. Maybe it doesn't sound as interesting as it is, but it's really pretty fun. Okay, so that's it. I wanted to close with just by saying that there really are beautiful animals, exquisitely adapted to living in the deep sea. Much of what's going on in the deep sea we really don't understand in terms of processes and importance for the welfare of human beings even. And we also have resources that are very valuable that we may wish to mine, if not in this century, somewhere in the future. And so this is a real opportunity. The mining industry gives us an opportunity to think really critically about what questions we need to ask, what answers we need to get, and figure out how we're going to go get them. So thank you. That's my story. You're my question taker.
Thank you very much for your fantastic presentation with beautiful stories and pictures from the deep ocean. So now we'd like to open the floor for any comments or questions. Oh, I think there's one in the back. He's kind of have it like half raised. Yeah, there we go.
Hello, thank you very much. This is beautiful. I have a question concerning those black smokers. Do they die? Can they die as systems? Because then that would, be a really ecologically interesting thing, but also for the mining.
Yes. Yeah, that's a great question. They can die. So it depends on where you are on the mid-ocean ridge. So the first fence found on the East Pacific Rise Galapagos Spreading Center in the Eastern Pacific, that's called a fast spreading center. And so the chimneys you could never mine there because the chimneys don't get big, the deposits don't develop over time because it's highly volcanic. And so they get overrun by lava. So they can die that way, but they just get overrun. Even there, they can die because they get clogged. They get clogged with minerals, and then the fluid has to come out somewhere else. And ultimately, you could quench the magma chamber, but that's in geological time, not in ecological time. So yes, on the East Pacific Rise, those things blink on and off, and it's really interesting to think about the connectivity, how populations are maintained, where the sources and sinks are for the larvae of these things. Almost all the invertebrates that live at a hot spring have a larval stage that goes in the water column. On the mid-Atlantic ridge, the reason that tag mound is of interest is so big because it's been there for more than 100,000 years. So it's just accumulated over time. So it takes a few thousand years to get a deposit that's old enough to mine. So the mid-Atlantic ridge is colonized by shrimp. The idea of an adaptation to being in a the East Pacific rise animal's going to recruit faster. There's a succession on the East Pacific rise, so the first thing comes in is tube worms, and then the mussels come in, and then the clams come in. So we know that pattern of succession. On the Mid-Atlantic ridge, we've not had a volcanic eruption that has covered over an active vent site. So we don't know how that succession works, and I don't know what will happen. Yeah. So they can be clogged, they can be overrun with lava. Yeah, they're ephemeral to a point. Depends on where you're at. In fact, maybe a good point is Nautilus Minerals. One of the reasons they like the Saguaro 1 site is they think of it as a waning site because there's so many dead chimneys, and so the dead chimneys don't have the animals with the symbionts. Instead, they have other animals that are old and slow growing.
Over here.
Over here.
Yeah. In the back.
Oh, yes, sorry, thank you.
So you just mentioned it very quickly that those black smokers and related deep vents are also implied for the origin of life, and that makes it super interesting for me at least. So do you know anything about how quickly genetic material or genes are basically spreading over those systems? Is there an active exchange of species between, let's say, very remote vents that are in similar conditions?
I don't know about microbes. So microbial biogeography of vent systems, there are some people thinking about that. Anna Louise Reisenbach has done some work on that. But for the invertebrates, there's a biogeography, and it's kind of by basins or part of basins. So there is a lot of mixing, like on the Mid-Atlantic ridge. Of course, it depends on the resolution of your genetic marker, and we start out with really low resolution markers, but maybe as we get better and better, or start to do larger parts of the genome, we'll be able to parse the populations better. But right now, for hydrothermal vents, for a lot of species, the dispersal corridor seems to be quite long.
We have one in the back.
Yes, thank you.
May I know, do you know any research team or any roadmap for building a real-time underwater wireless control vehicles? Because I know there is a team working in CAUS, they're working on underwater wireless video transmission. Maybe there is some chance for collaboration in the future.
Yeah. So there's a lot of interest in having good acoustic control. So acoustic control, not radio, and trying to have some repeaters. So I guess I forget how they want to do it, but they have, maybe it's more for the navigation, right? You have a navigation buoy, and you need your vehicle needs to be able to drive. You want to have the ship leave it or send it out from shore, and then be able to know where it is. So you don't have GPS underwater, so you have to ping to a surface buoy and then ping to a satellite. I think for Sentry, they really are trying, and it sounds like maybe not so far along the lines, they're not trying to get video back, but they're trying to get better able to program or reprogram, make bigger adjustments to what it's doing on the seafloor. Alvin, we were able to tweet from the seafloor using an acoustic modem, and they would like to get to a point of having video coming up through some optical means, but they haven't. That's a long ways away. So yes, it'd be lovely to hear about those kinds of things. That's the kind of thing that your folks here must just love to get involved in and figure out how to make it work. Yeah, that'd be great.
One down the front.
Yes.
Thank you. Very similar questions about autonomous vehicles. Can you say what will be future of these vehicles in 5 or 10 years? Can they go very deep, 10,000 meters? Do they need does GPS work so deep, and what are difficulties?
It's not, navigation is not by GPS. I am not an engineer, and I'm going to say all the wrong words, so the engineers in here just, kind of close your ears. But I know for Sentry, just to be able to do that 1 meter above the bottom, they had to buy a special navigation system to do that, to have that good control, to know where you're at. Navigation is key. If you get data and you don't know where it came from, it's useless. It's really not well, it's not useless, but it's not very helpful. I actually got to read a proposal, so I do know what the future of Sentry might be if it gets funded, but I can't tell you. And it's really cool, but sorry, in 6 months. No, I don't. I think for me, so if you're asking me what I would specify, I want, and I'm going to give you one of my proposal ideas. No, I'm not going to do that. Sorry. Good question.
Hi.
I hear it. I hear you, but I don't see it. Okay, thank you.
Hi. I have a question about the evolution of these animals in these habitats. I don't know if you have an answer for that, but because a bit earlier, a friend of mine, I'm a marine scientist, and a friend of mine asked me, why don't we find animals in this area that are more original than their counterparts in other parts? Like we find crabs, they are crabs.
Yeah.
We find mussels, and they look like mussels, but Imodulus is a mussel.
Yep.
We find shrimps that looks like shrimps. So why don't we have some totally different animals in this area, except for Riftia, maybe, that is really weird?
There's shallow water Riftia as well, but so the answer, as I understand it, and I could be corrected or be wrong or be right, is that there were major anoxic events that reached to the deep ocean, and so you reset the clock. The shallow water roots, ancestors were able to survive. They weren't involved in those anoxic events, and so everything died, and the clock got reset. So the things had to come back in and invade. So to my knowledge, there's really not much at a hydrothermal vent that's ancient. We thought originally that maybe the barnacles were ancient lineages, and that's not so. So that's not a good answer. I'd have to go Google some to get a better answer. But I think it's the anoxic events. Does that sound good? Okay. Anything else?
Any other questions? Oh, one in the middle.
Yes, I see this one. Thank you.
Thank you. I'm curious about the negative impacts of the mining. If you're basically taking out a lot of highly concentrated ore or disturbing the seafloor, are there some possible negative events that you could trigger that could be very far-reaching?
If we just talk about the seafloor massive sulfides, I suppose if the toxicity is of concern, but whether it would have an effect that would be really over a huge scale for seafloor massive sulfides, that's harder to see. You're going to have, if you mobilize the metals that become bioavailable, then you're going to have toxic effects on the larvae and in the water column. But my sense is it will be transient. The mine sites are 3 to 5 years, 10 years at most. So transient on that time scale, which is worrisome, but maybe not so bad. It's one of those things where you really have to just start monitoring, because sometimes it's just too hard to model. Right now, we don't even know what the mining tools are going to be like for some of this. So maybe transient. The vent animals would do fine because they're exposed to metals all the time. It's the animals that surround that are the concern. The sediment plumes that might get kicked up and formed are a concern because they will bury things or clog filter feeding apparatus. There might be engineering solutions to that, where you put a shroud over the cutters and somehow minimize the impact. And then again, for hydrothermal vents, the vent animals might come back. We're actually arguing that vents should not be mined. The active vents should not be mined. That's really the position our science recommendation is taking. Don't mine the active vents, but you can mine around them. I mean, yeah, you can mine around them just as long as you don't have an impact on them, because they're just too special. They're, you know, place that's the origin of life. You really shouldn't be mucking around with that, or could be the source of the origin of life. Other impacts. So if you think about manganese nodules and cobalt crust, there is where it really gets bad. So manganese nodules, if you mine them, you're taking a lease is the size of Austria, so it's the size of a country. Within that, they might take 30% of the nodules. In fact, I was talking to one of our colleagues here. It's like, well, I really can't tell you. I know if you take a square meter of mud and its nodules, I really don't care. Really don't care. You take 10 square meters, I don't think I care either. I don't know at what point I do care. So do I care if an area the size of Austria is disturbed by a mining event, or some fraction of it? I don't have a sense of that. The scales are not in my head, and I don't think they're really in any of our heads, of how this matters in terms of carbon sequestration, productivity, secondary productivity, those things that matter, and where are the tipping points. I have no idea where the tipping points are. That's those are the scary things. And then again, the cumulative impacts, and how do you manage on a regional scale? The lack of knowledge is huge, and so we're happy. There's ways to go get some of this knowledge, but it's going to cost money and time. Manganese nodules, they take millennia to form. The animals that are specialized to live on manganese nodules, they're not going to come back. So there's some ideas of, well, put concretions down, put concrete down, or the tailings down. But when you do, you want to test that, right? To test it, how are you going to do that? It takes a century, decades to a century to know if you've got a successful recovery. So we're grappling with those things. We're not saying, no, you can't do it, but like, help us. We don't see how you could do this for manganese nodules. And it might be it's okay, but we just don't. How are you even going to know if it's okay? That's the monitoring. What metric? If you go to any deep sea biologist and ask, well, what do you measure to know if you've had an impact on the seafloor? How do you know which one you're going to measure? Which one can you afford to measure? At what scale? And what spatial scale and what temporal scale? Anyway, yeah, you're asking, this is what all the things we think about all the time. Cobalt crust, you're going to scrape off all those old growth corals on a seamount. What impact does that have? The real naysayers could say you just shouldn't do it, but it certainly bears thinking about what really, how bad would it be? How will we know how bad it'll be? Good questions.
Okay, and one last question down on the right.
Okay, so how did the humanity benefit from what's been so far done with all these deep sea exploration? Mm-hmm. What are some examples?
Well, from the hot springs, there are, that's high temperature, high pressure, and so the enzymes of the animals that live there and the microbes that live there are adapted to high temperatures and pressures. They can be used in industrial applications for us, and they are used. A good example is the TAC enzyme comes from hydrothermal vent, or one of the enzymes used in molecular biology. I don't think it's TAC, but one of them comes from hydrothermal vents. So it's used all over the world in molecular biology. There's an enzyme that works at very cold temperatures and high pressures that's used in industry. There's one that works at cold temperatures that's in a laundry detergent. This is not my expertise. I wish I should be prepared with a better answer for this than I am. Some of it is maybe into the future. I gave a little on-air interview this morning, and one of the things I mentioned, we don't know really how it will work in the end, but we found a snail with a scaly foot, and it was armored with metal. Colleagues at MIT, material scientists, looked at how that metal was integrated with the tissue layers and the shell layers. And they wrote, I think it was a Nature paper, about a new kind of biomimetic armor based on the structure of this. Sponges are a good example. There's a big European Union program on sponges, and I learned that sponges, the silica skeleton is a good substratum for cell culture. So you can start, if you need to form new organs, you can form it around these silica structures. I'm not exaggerating, I'm just probably misstating the whole science of it, but it's this idea that you can use the skeleton as a lattice framework for tissue cultures for growing organs. So there's real interest in that. So it's kind of all over the place. There's cosmetics, it's way too many things. And then, way too many things. And then just knowledge, science, the idea of thinking about how life originated, how our animals adapted to extreme environments. They teach us a lot about what's happening in shallow water.
Let's give Professor Vandover one more round of applause.
Thank you very much. It was a great pleasure.